see who's here. One, two, three, four. Right. Anybody got any questions on the homework that they had? It's quite complicated, I think, now because you're at the point where we're doing lots of mixture of things, and some of these booklets get a bit more challenging. But I already have some questions. One, two, three, four, seven, eight. Yeah, sure. Um, start with that. From the year seven booklet. Um, okay, let's have a look. These are just very random mixture of questions, so they might not always cover things that we have done in the lessons, but it's just a bit of a mixture of things. Any other questions, then feel free to ask from the homeworks first. Um, okay, the diagram. So what we said, one, two, three, four, seven, eight, shows a fish called a cod. Name one part which it uses to move through the water. You want to say either the tail is probably the simplest answer, really. That's what helps you move, doesn't it, through the water? The fish has gills. What are gills for? Gills for fish are for breathing or to take in oxygen. I think that's a better answer, actually, because technically you don't breathe in the same way. Okay. Look at the shape of the fish. How does the shape help it move easily through the water? You want to say something about like streamlined, a streamlined shape. Like if a swimmer has a streamlined body. I mean, they tend to all have streamlined bodies, swimmers, because of the swimming, and it helps them swim. That's what we want to say. This shows the food eaten by cod. What is the main food eaten? I don't think I need to answer that, but it's surely the small fish. And a food chain which ends with cod is that a food chain is basically the chain of things that eat other things. An arrow in a food chain means eaten by. So a tiny plant is eaten by a tiny animal, which is eaten by a small fish, which is eaten by cod. A producer in a food chain is always the first thing in the food chain. It's always a plant, basically. Um, so a tiny plant. Going through the year seven booklet at the moment. If you have any questions from the other ones, then please feel free. I don't think there's many people are going to be on today, but if you do, then just let me know. Oh, the printings are funny here, isn't it? A cod eats other animals which live in the sea. What is the name for an animal which catches and eats other animals? It's called a predator. A predator is one that eats prey, eats other animals. One, two, three, four, seven, eight. So two. The drawing shows a fish tank, another fish. Um, it has some waterweed and some fish in it. During the day, waterweed give off bubbles of oxygen. So the reason why, if you ever look at a fish tank, there needs to be some plants in the fish tank because, as you know, plants do something called photosynthesis, which releases oxygen. Now, without the plants, there wouldn't be enough oxygen in this tank for the fish. So, two ways in which the waterweed helps the fish to stay alive. Um, one is it provides oxygen, and also, it will be some of their food, as in they will be able to literally eat some of it. So it will give them food as well. Um, Booklet three. Yep, I want to come to number seven in that in a moment. Yep, sure, sure. Tick the boxes by three things which both fish and water weed do. They both use energy. I'm sure that's correct. They both reproduce, animals and plants reproduce, and they both grow. That's got to be the answer. They don't both move from place to place, do they? They don't both eat. A plant doesn't eat in the same way. They don't. A fish doesn't make its own food, whereas a plant does. So it's just understanding the differences. Um, three, four, seven, eight. Three. Some people have unbalanced diet. This can give them problems with their health. Draw one line. Right. Too much sugar is probably tooth decay. I'm just going to assume that's correct. Too much fat might lead to heart disease. Carbohydrates are the kind of food that help give us energy. So if you don't have enough carbohydrate, you might not have much energy. Proteins are the foods that help us grow. People who like go to the gym and build muscles eat a lot of protein because it helps their muscles to grow. So without it, you'd have quite poor growth. Why eating lots of chips every day is bad for your health? I'm going to probably say that they've got a lot of fat in. That seems like a sensible answer. Too much fat, yes. They can make you overweight, that kind of thing. Okay. One, two, three, four, seven, eight. Andrew puts his rabbit's cage on the grass. A week later, the grass under the cage had turned yellow. One reason why it turned yellow. Well, surely, what do all plants need? They need light, don't they? And so, if you suddenly put something on top of the grass, it's not going to get any light, and that can cause it to basically change color. Um, okay. Andrew wanted to test why the grass had turned yellow. He put two sheets of plastic just above another patch of grass. One was black and the other was clear. Well, obviously the black one isn't going to let any light in, whereas the plastic one probably will let light in. A week later, the grass under the black sheet was yellow, the grass under the clear sheet was green. Explain why you use the clear sheet as well as the black sheet. The reason you need both of these is you're trying to compare um, um, the two. It is needed as a comparison or to make it a fair test. Because without this, it's not obvious why this has turned yellow. Because if this wasn't there, maybe there was some other reason they turned yellow. It wasn't to do with the light, it was because somebody came along and painted them yellow. But with the clear sheet, we can now compare light versus no light and see the difference. That's the point. He left the black sheet there for several more weeks. What happened to the grass under it? Probably died. Um, yeah, because without light, eventually a plant will die. Two things which both rabbits and plants do. They definitely grow, they reproduce. Um, is it going? Oh, it's only two. Sorry, I thought there were three. Only two. So it's only those two. Um, and then seven and eight. Oh, look at that. A wind-up toy, dog, a rabbit, and a rose bush. In what way are the toy dog and the rabbit similar? They can both, well, they can both move from place to place because a wind-up toy moves, doesn't it? But it doesn't eat or grow or reproduce, right? So moving from place to place. Two ways in which the tiny dog is different. It cannot grow and it cannot eat. So I think those two go there. Two ways in which the rabbit is different from the rose bush. The rabbit can move from place to place, but the bush can't. And the rabbit can eat, but the bush can't. The plant can't eat in the same way. And then lastly, eight from this one. Changes to the environment sometimes harm plants or animals. Use only the worst places. Rivers may be polluted by sewage and fertilizer. I'm just trying to check that's the answer. Fertilizer, basically, we put it on plants, but if it rains and stuff, it can get washed away and go into the rivers. And if it goes into the rivers, it can cause problems. Um, booklet three, I've got some questions from booklet three, but you have to specify the booklets the question numbers. I'm not going to do the whole thing. River pollution harms and sometimes kills fish. Oops. Oh God. Oh no. What's happened here? Right. Uh, Fish, I'd have thought. What else can it kill? Insects? Yes, anything animal related. Oops. Hedges may be dug up to make more farmland, must be. The reason you dig up a hedge is so that you have more space to grow things. Um, Digging up hedges destroys the homes of some insects and birds, I think. They're the things that live in hedges, right? Potentially. Sorry, I'm not writing all these in. But that is question eight, all done. Right. Booklet three, I seem to be question seven, seems to be the problem. Let's have a look at that. Oh, so much going on here. Sally investigated how the human body digests and absorbs starch. She used saliva to digest the starch. Your saliva contains an enzyme called amylase, which is used to digest starch. To model digestion, she used special bags made from a semi-permeable membrane. Semi-permeable means that some things can go in and out of it, some things can't. These bags have lots of very small holes, therefore small things can get through, big things can't. Sally sets up the equipment. Look, there's one special bag in each, be a special bag. Um, so we have beaker A. which has a bag which has got some saliva and some starch in it, and the water around the outside. But all of them have got water. This has starch only, and this has saliva only. Okay, so just so you understand what's going to happen here. Saliva contains something that breaks down the starch, right? So any starch in here will get broken down into something smaller, and then it will probably go out of there once it's small enough into the water. Now, in this one, um, nothing will go out of the water because there's starch in here, but it's too big to get out, so it'll just stay in there. And saliva, I'm not sure what will happen, but we'll see. I think it'll just stay there as well. Well, we'll find out. She keeps the water in the beakers at 37 degrees C, which is the body temperature, which I think is the answer to the first question. After 20 minutes, Sally tested the contents of each beaker and bag for starch and sugar. Here are the results. So, was starch found in the bag after 20 minutes? In beaker A, yes, there was still some starch in the bag. There was some sugar in the bag because the starch gets broken down into sugar. Um, was any starch found in the water? No, because starch doesn't get out. Was sugar found in the water? Yes, because the sugar is small enough to get out. Okay, that's what's going to happen. In beaker B, was starch found in the bag? Obviously, because it starts in the bag, but then nothing gets out, right? And so there's no sugar because it's not broken down. There's nothing to break it down. There's no starch in the water because it's too big to get out. There's no sugar in the water because there is no sugar in bag B, because it's not broken down. And in bag C, nothing happens because there is no starch. So it's just nothing. Okay. So always with these questions, take your time to understand the information, right? If you just jump into the question, sometimes it's really hard to then go backwards and understand it. Why did we keep the water at 37 degrees C? Because it is your body temperature, and we're trying to replicate what happens in the body. Okay. Explain why sugar was found in beaker A. The reason sugar was found in the bag in beaker A was because the starch is broken down into sugar. Okay. Starch was not found in the water outside the bag in any beaker. Why? Starch is a really big molecule. It doesn't get through the small holes in the bag. So it just doesn't get out. Why did Sally set up beaker C? Um, I think it's for a fair test. Oh no, for a control. So that's the correct answer. A control means something that you compare to, basically. So it's always so you always do one that's nothing. So you just compare to it. Um, they used diagram to show what happened in her investigation. Oh God. Um, which diagram shows the results of beaker B? So beaker B was the one where there's starch inside, but it's too big to get out. So isn't it P? Yeah, P, because there's only starch, nothing broken down, and then it just doesn't get out, so it just stays there. Um, so P, which shows A. In A, what happened was some of the starch got broken down into sugar, and some of the sugar then moved out of the bag. So we should have starch, there's starch on this side, and some sugar because it's broken down, and some of the sugar has gone across into the water. So R must be what's happening in beaker A. What does saliva contain? It's called there's enzymes, but let's try and name it. The name of the enzyme that breaks down starch is called amylase. Oh, so sorry, I muted myself somehow. Anyway, sorry. The yes, the answer was five for the first one. For the next one, what happens to the oxygen level as you travel downstream? Um, Downstream is that way from Pine Bridge, and then it increases, as you can see there. Okay. Um, Trout only live in water with oxygen levels higher than 20. How far downstream would you like to find trout? So what we need to do is get above 20 on this axis. So there's 20. So we need to get to here before we have enough oxygen. So 2.5 meters, or further than 2.5, not meters, kilometers. Um, so 2.5 kilometers. You've got to say the unit. And then the scientists collected samples of the river animals found at different places. Okay, right. Trout only live in water with oxygen levels higher than 20. Give the name of one other animal that only does that as well. Now remember, we've already established that the oh God, where is it? It was 2.5 kilometers um, from the bridge. Oh, but on this side of the bridge, 20 happens here. Okay. So anything less than zero point 0.5 or zero point 0.5 minus zero point 0.5 would be fine. So I think any of these are the answer to the question. Um, yes, stonefly nymphs, mayfly nymphs, caddisfly larvae. Um, Surely freshwater shrimps. Oh no, freshwater shrimps, no, because they also are there. Um, so yeah, basically that one. And the one below it is fine. Name two animals that are only found when the oxygen level is below 10. Right, below 10, you have to look at two graphs here. So you see below 10 is there. Now that only happens when you're very close to the bridge. So 0.51 from the bridge. So 0.51 from the bridge is only these, well, not even that one because that's got there as well. So I think it's only these sludge worms or blood worms. Um, and rat-tailed maggots. So any of those three would be the answer to that one. Um, in the river, trout are predators. Near Pine Bridge, the number of trout decreased. One reason why pollution may cause the trout population to decrease, because there's less oxygen, less food available. Um, yeah, if there's pollution, pollution generally means that there's less food to eat. There's less oxygen. That's why their number might decrease. Less oxygen is probably a sensible answer. Okay. Um, Right, and then that was one, and then you had said 2A and 2D. One evening, Jenny and Lee ate chicken sandwiches which had been in their school bags all day. There were harmful bacteria in the food. The next day, both girls became very ill. They gave them antibiotics. The graph represents how antibiotics affect the number of bacteria in the body. Okay, so sandwich is eaten, the bacteria starts to increase, the antibiotics are started, and then gradually they start to decrease because antibiotics start to kill them. Why did they not become ill until the day after eating the sandwiches? Probably because the number of harmful bacteria wasn't very high until the next day. You see what I mean? It was quite low initially. The next day, there was a lot more of it. You see what I mean? So it increased very quickly over the course of a day or so. Um, After taking the antibiotics, Jane was explain why she got better. The antibiotics killed the bacteria. Leah should have taken the antibiotics for 8 days. She felt much better after 5 days and stopped taking them. 2 days later, explain why Leah became ill again. Well, after 5 days, there were still some harmful bacteria in the body. So just because you felt better doesn't mean that you've killed all of the bacteria. You felt better, but there's still harmful bacteria. So you want to say that there was still harmful bacteria in the body And what did that harmful bacteria do then once you stopped taking the antibiotics? They then multiplied and became a really big number again. And that's why you became ill, which is why you want to make sure that if you are on antibiotics, you finish your course of antibiotics. Um, because that makes sure that even if there's a little bit left, you kill them all. Otherwise, they will multiply and you could get ill again. That was that. Um, um, the low temperature, just so you know, for the answer to this final question, it stops them multiplying. That's what it does. When it's cold, the bacteria don't grow as fast. That's why we have fridges and freezers, basically. Um, and anything else? There was 2C done. One, 2C, 5 part 2. Is that what you said? I think you said that. Um, um, the percentage of black moths is shown on the graph. Continue the line to show the percentage of black moths change between 1980 and 2000. So black moths presumably just went down. So these numbers have to add up to 100. 10 add 90, 50 add 50, 78 add 22. And so the percentage of black moths was 90 in 1970. Then in 1980, I guess I presume that's 1980. What else could it be? It was 50. So 1980, a mark at 50. Is it 1980? Let me just check the answer to that. 1985, apparently. Okay. So in 1985, it dropped to 50. So you want to continue that to there. And then in 1990, it was 22, which is about there. And so you continue this like that. Okay, that's how you continue the line. Um, um, and I think that's it. Let me have a look at question three. My hesitation at the moment is the fact that you are not asking all the questions together, which suggests that you're looking at them as we speak. Um, I'll just tell you this in words. In the wall of the trachea, there are pieces of stiff material called cartilage. Why is it necessary? Is to stop it from closing and to keep it open. It's like a protection, having a solid layer around it keeps it open. Okay, it prevents it from closing. Um, yeah, to keep it open, I think is the best answer. B. I am not going to answer that question because you should know what gases are involved in terms of your breathing. Um, the reason why it's easy for gases to pass across these is because these alveoli have thin walls. That's what we want to say. Um, and the function of this cilia, we have cilia in our throat, which helps to move mucus back up our throat. So when you breathe in stuff, you don't want to breathe in anything that's bad, I suppose. Um, and it gets stuck in the mucus. You have mucus in your nose and throat, but then you have to bring that mucus back up, and these little hairs help to move the mucus back up your throat. Um, what effect does cigarette soap have on the cilia? It destroys them. I think that should be okay, or it just stops them from working. Give the name of a cigarette which causes addiction. That's called nicotine. That causes addiction in smoking in cigarettes. Is that everything? Amazing. Right. Great, good. Well done for coming anyway. It is important that you guys come and ask for help. Um, because the more you do that, the more you improve. Right, I think I'm going to leave it there. And should I do 3 ABC? Yeah, I did 3 ABC, didn't I? Yes, I did. I'll be back in about 20 minutes because I might have some year 10 students arriving for some assistance on their homework. Um, yeah, it is being recorded. It ends up on the YouTube and there's a link that gets emailed to you. So you can watch it back, but it won't be on there for a few hours because it takes a while until I finish. So yes, I'll be back. Right, not many people here. I just want to see if there's anybody's got any questions. I won't stick around if there's not any from the year 10 homework, or any other questions, I suppose. Won't keep you for long. If you do have any questions on this homework, for which you can actually find the mark scheme quite easily, then let me know. I have been through some of the Key Stage 3 homeworks, but if you do have any questions from that, then let me know. Otherwise, let me know if there's any questions from the year 10 homework. We did a recap of biology things. We will do a similar thing for chemistry next lesson, and then physics, and then there's a test actually at the beginning of July before we close. So if either of the couple of people that are here have any questions, then tell me in the next few minutes. Otherwise, we will end this early because the purpose of it really is to ask some questions on any of the homeworks that you have. I'll give you a couple of minutes to decide, and if not, we'll call it there for today. Hmm. No, I'm not going to do all of the questions in the year 10 booklet. The purpose of this session is for you to have attempted the homework and then to have asked for some questions that you have tried. I'm not going to do all of the questions in the year 10, especially questions that are like, what is that structure? So if you've attempted it, then you should be able to do some of these questions. But I presume somebody will come on shortly and ask some questions from this, and therefore I can do some of them. Um, Shreyans usually asks some questions. Oh, good. You found the paper quite easy. Good. Yes, because it should have been quite easy in theory, because it's a foundation level paper, which you won't end up actually doing. So, on that basis, given that there are no specific questions, I won't carry on because only two or three of you here today. I think I will leave today's session. Remember, the purpose of these sessions is for you to have a go at the homework, come and say, didn't understand these specific questions, and then you can, and I'll happily go through them. I just want to encourage people to attempt things and then ask for help. That's what we're trying to do. Um, a plant revision session. What I'll do, I'll read through a few slides if you like. Sure. Um, so, um, well, we could help us inspire. Sure, happily. Um, so these notes, which you technically do have access to somewhere, but if you don't, then let me know. But the a little bit of a plant revision that has been asked. But if you do have any homework questions, please do ask specific questions. Um, I'll just read through these. I won't keep you here forever. But basically, this is important, which is the if you look at a leaf sideways, this is what you see. Well, you don't see, but you would see if you had zoomed in on it. And that are the different layers that you find in it. Now, the important thing to recognize here is that this layer called palisade mesophyll, if you look at the picture here, is what contains most of the chloroplasts, which means the majority of the photosynthesis happens here, which is because chloroplasts are where photosynthesis happens. Now, what does photosynthesis require? It requires, so there's an equation for it, carbon dioxide, water, and it produces glucose and oxygen, right? And it also needs light. So there's three things that we need for photosynthesis to begin. We need some light, some carbon dioxide, and some water. Where does the light come from? The light comes from obviously the sun or any light. Now, the this layer, this epidermal tissue, the thing about it to know is that it is see-through. Light can pass through it, which is important because the light needs to get to this sort of second layer where all the chloroplasts are. So that covers that bit. Um, Question 3.3, I will do that in just a second. Yes. Um, So that's where the light comes from. Where does the water come from? The water comes through these cells called xylem. Xylem cells transport water from the roots up to here. And where does the carbon dioxide come from? It comes in through these stomata at the bottom, which are open and closed by guard cells. And conveniently, we have a layer of spongy mesophyll, which has got loads of gaps. You can see the gaps in this picture, which allows the gases to get through to the place where all the photosynthesis is happening. So this picture kind of summarizes a lot of the stuff you need to know about plants. I will come back to that in a second, but I just want to answer the question on the homework. Question 3.3. What's it asking? I don't know. Whoa. Okay. Right. So, quite a complicated question, actually. The only thing you're meant to know about yeast is that it is something that can do a process called anaerobic respiration. So, yeast is, and the benefit of this is that if you put some yeast in a place where there is with no oxygen, so it respires anaerobically, it produces ethanol and carbon dioxide. Now, ethanol can be used to make alcoholic drinks. Hence, yeast is used in the brewing industry. That's basically one answer to that question. So, yeast is used because it respires, when it respires anaerobically, it produces ethanol, which is needed for the brewing industry. The other thing that is helpful is that yeast is used in making bread, and the carbon dioxide helps to make the bread rise. When you put bread in the oven with yeast, it rises up. It starts off flat, it goes up. And so that's why it's used in the baking industry. So in the brewing industry, the ethanol from anaerobic respiration is used to make alcoholic drinks. And in the baking industry, the carbon dioxide that's produced is used to make the bread rise. Um, 6.4, yeah, 10. Okay, fine. I'll get to that. Infected mosquitoes landed on socks. I probably should read the whole question, but I should be okay. Infected mosquitoes landed on the socks three times more often than uninfected mosquitoes. Oh God, I need to understand what's going on here, don't I? Infected mosquitoes, socks worn for a day. Socks worn for a day. Okay, uninfected mosquitoes. And they recorded the number of times they landed on the socks. And it seems that infected mosquitoes landed on the socks three times more often than uninfected mosquitoes. How can this be used? I suspect the answer is that you should change your socks regularly. Because if you change your socks regularly and don't wear socks uh, worn for a day, then fewer, then the mosquitoes won't land on them as often, which means that we won't spread. Remember, mosquitoes have to bite you to spread it. And if they're landing on your socks, they're more likely to bite you. If your socks are a day old, they're more likely to. I think that's going to be the answer. Let me double check. Hmm. Oh, yes. Oh, yes. Wear clean socks, change socks regularly to reduce the chance of attracting mosquitoes is an answer. So change your socks regularly to reduce the chance of attracting mosquitoes is a sensible answer. Um, yeah, that's I think that's the best answer. There is another answer, which is that we can use worn socks to attract the mosquitoes and then kill them. But I think it's better to say if you change your socks regularly, you've got less chance of attracting them. So that's 6.4. 7.2. Figure 9 shows some of the apparatus used to measure the rate of photosynthesis. Yes. Um, the rate of photosynthesis in the pod is affected by different colors of light. How could you investigate this? Right. So, um, what you would want to do is, so different colors of light. So set up a lamp, um, and then you need a fixed distance. So let's say, um, 30 centimeters from the pondweed. And that is something you would control. So it says variable you would control. That distance should stay the same because you're not trying to change the intensity of the light. You're just going to change the color of the light. Okay. Then what you would do is you would um, turn the lamp on with one color. So let's say with one color, turn it on, and then measure the number of bubbles produced in a minute. So you have to set a fixed amount of time, which is also a control variable. So count bubbles for 1 minute, let's say. So that shows you how the photosynthesis is happening with yellow light, let's say, how the rate of it, the number of bubbles produced, the more bubbles produced, the faster it's happening with that color of light. Again, that time is a control variable. It should be the same every time. Then what you would do is you would change the color. The way you do that, you could say by using a different light bulb, right? You could change the bulb and that'll change the color. And then repeat the experiment and count the bubbles again. And then what you should probably do as well is at the end do the whole thing again. So do it, let's say, for each color, do the experiment 3 times and calculate a mean. That's always a mark for one of these questions. Sorry, I've written this really badly because zoomed out. Um, so yeah, it's basically an investigation how you could figure out how the different colors of the light affect the number of bubbles produced. So you would set all of this up, start with one color, count the number of bubbles in a minute, and then change the color, count the number of bubbles in a minute. That kind of thing. Um, 2.1 and 3.3. 3.3, I think I might have just done, which was this one. So in 3.3, the idea was you're meant to recognize that yeast is something that we use or we learn about as something that does anaerobic respiration. So anaerobic respiration um, is when we have respiration without oxygen. And when you do that for yeast or plants, the products of anaerobic respiration are ethanol and carbon dioxide, whereas for animals, it's lactic acid, so it's a different process. So what you'd say here is yeast is used in the process of anaerobic respiration. The ethanol produced is used to make alcoholic drinks, which is why it's used in the brewing industry. The brewing is the drinks making industry. And then also, yeast can be used in the baking industry in the same process because the carbon dioxide that's produced is used to make the bread rise. That is the answer to this question. It's quite a convoluted question. Um, and 2.1. Um, how do you do this? Okay, I think what you need to do is get your ruler out because it's hard for me to do this because I can zoom in and out. But get your ruler out, measure this, right? Let's pretend, just for the sake of argument, this is 1 centimeter when you measure it on your page. And then measure this bit here. And let's pretend, for the sake of argument, that that is, what is it going to be roughly? One, two, probably about 3 centimeters roughly, or three of these. So however many of these are in this space, and then times that by 2. But then give it to the nearest micrometer to round it. So I'll tell you what the actual answer is. Should you be interested? 8 micrometers. So I presume this is close to four of these. If you actually do this, there's probably about four of them. One, two, three, four, roughly. Which means that if each one of these is 2 micrometers, altogether that's 8 micrometers. That's the way you do that. Right, that's um it measures 3.6 centimeters. Okay, but what does this measure? Um, also, what's 3.6 times 2? The answer's 8. So, yeah, the answer is 8, according to the mark scheme I'm looking at. So I assume that yes, this might measure 3.6, but they need to figure out what this is. That might not be 1 centimeter on your page. Um, so it's essentially how many of these fit into there, which should be about four of them, right? So yeah, it depends on. I can't do this because I can, as I say, zoom in and out, which ruins it. I can't use a ruler um, to do this. But yeah, the answer is 8. Um, the answer is 8. It measures 6 or 7 micrometers. Um, the width of the cell. I mean, is there any way that I can do this? Oh, there might be. Let me see. Okay. Okay. Very clever what I'm doing right now. Right. So you see, that's almost four of them. Slightly less than four of them fit into that space. Slightly less than four. Let's call it 3.8 of them. Um, but then 3.8 or 3.7, whatever, times 2, because each of them is worth 2 micrometers, rounds to 8. See what I mean? Because the question is to the nearest micrometer, so that's why it's 8. Yes, if it makes 0.9 centimeters, you have to find out how many go into 3.6 of those. So if this is in centimeters 0.9 and all of this, according to you, is 3.6 centimeters, then, I mean, that's four of them, isn't it? 3.6 divided by 0.9. That means four of them fit into there. Each of them is 2 micrometers. 8 is the answer. Hmm. Okay. Anything else? So somebody asked me to just talk about some photosynthesis or plants stuff, so I'm going to carry on with that unless there's more. Um, right. So I explained that. The other bits of this topic are knowing what phloem and xylem do. Xylem transports water. Phloem transports glucose. Not a huge amount else to know about that. Guard cells are, as I say, the cells that can open and close depending on um, whether or not they need to be open to let gases in. When they're open, we can photosynthesize. When they're closed, they're not. It's a bit more complicated than that because these only open when there's plenty of water available because that makes these cells fill up with water and then the gap opens. Whereas when there's a lack of water in the plant, they kind of collapse and the stomata closes to prevent too much water being lost. Because remember, the stomata is not only to let gases in and out, it accidentally lets water out as well. Um, because diffusion works that way, so it just happens. Um, that is that. Anything else? Transpiration, I want to leave for now 'cause it's complicated. That is the equation for photosynthesis. Um, we need to know the symbol equation as well. And then this is quite important. I mean, I see these questions a lot, and they came up in the GCSE exam just now as well. Um, how plants use glucose. So yes, you know that photosynthesis makes glucose for a plant, but you know what it then uses for, because it's a very common question, worth like 3 or 4 marks usually. Why do they use it? So the simplest answer is because they respire, and that respiration releases energy. Um, so that's that. Um, but then there's other answers which sometimes are less obvious. So cellulose is a substance that is used to make the cell walls. Your cell walls in a plant cell are made of cellulose. Glucose is needed to make that cellulose. Glucose can be converted to amino acids, which is used to make protein. Remember, proteins get broken down into amino acids. So amino acids can be built up to make proteins again to help it grow. And it also stores glucose as starch. Because it needs it for when it's nighttime, for example, because it can't make more glucose, it stores some of it. That is a very common sort of question. The rate of photosynthesis is a slightly more complicated topic. The rate of photosynthesis is affected by three things: temperature, carbon dioxide, and light intensity. And the key thing about this, without me going into too much detail, is that all three of them need to be high to get the rate of photosynthesis to be high. It's not no good if it's just two of them. Um, so if it's really bright and really warm, then there might not be enough CO2. If there's loads of CO2 and it's bright, but it's not warm enough, photosynthesis won't increase. All three of them have to. And if one of them is too low, we call it a limiting factor. I've got a question. 7.2, I'll just keep skipping back to that. Was 7.2 the one I practical one? Mm, mm. Yes, it was. So I'll just repeat. I'm so sorry, the handwriting's terrible here. But this practical is usually, this is important, usually done like this, where you have a lamp and you shine the light at this pondweed. It's meant to be a lamp. And you, for about a minute, you count the number of bubbles because that's producing oxygen while it's got light. And then what you do is you move that light a bit further away, and you then do the same thing again, and then you move the light a bit further away, and you do the same thing again. You are measuring or investigating how the intensity of the light affects the rate of photosynthesis, because and as it gets further away, you find there's fewer bubbles in that 1 minute that you're counting for, or 2 minutes, whatever it is, as long as it stays the same. Um, but this question, and this is what these practicals do, and over the course of the next year, we will talk about this more. But the practicals sometimes give you a slightly different scenario, which is what this question did. It wasn't telling you about the intensity of the light, it was about the color of the light. So they're basically asking you, how would you do an investigation about how the color of the light affects the rate of photosynthesis? I will do this with an illustration to demonstrate this, um, which is that what we need to do is set up a lamp, just like before, and we count for a minute the number of bubbles. And that 1 minute is a control variable. It is one of the things that you have to specify that you control. It needs to be the same amount of time each time. So you do that for a minute, count the number of bubbles produced with yellow light, let's say. Then change the color of the light and do the same thing again. And the way you can change the color of the light is by using a different light bulb, for example, with a different color, or you can put a filter in front of it and that changes the color of the light. And count the number of bubbles for a minute and see if that makes a difference. Then change the light color again and count the number of bubbles. And what you are doing now is you are investigating how the color of the light affects the rate of photosynthesis. You see what I mean? You're seeing how the number of bubbles will change depending on which color light you're using. Things that need to stay the same are the time that you do this for and also the distance of the lamp from it. You are not investigating how the light intensity affects it, it's just the color of it. So that intensity should stay the same. So you don't want to change the distance of the lamp. That's another thing you would control. Yes, the distance of the lamp is something you should control, as in it shouldn't change, because this is not the same practical as the one you normally learn about, where you are changing the distance because you're investigating the light intensity. You're changing the light intensity. Here, you're not changing the light intensity. You're just changing the color. So keep the intensity the same, keep the distance the same, and just change the color. That is how you would do that. 9.1 has been asked as well, at which point I will probably finish after this. In 2014, there was an outbreak of Ebola virus. There's another outbreak of it now in Africa. At the time of the outbreak, there were no drugs to treat the disease, no vaccines to prevent infection. By March 2015, there was an estimated 9,850 deaths worldwide. It's an estimate. Why is it an estimate rather than an exact number? I mean, because we just don't really know. We don't need to give a measurement of the distance. No, you can just, well, you don't need to, but you need to be clear that it needs to stay the same. So I would personally say keep it at 30 centimeters from it and just explain that that needs to stay the same. But you wouldn't need to specify the number as long as you make it clear that that should stay the same. Um, why is it an estimate rather than, I mean, because we just don't know, do we? But how do you phrase that? Let me see what the mark scheme says. It says not all deaths are recorded. So you wouldn't necessarily record every single death. We wouldn't always know the cause of the death, right? I know there's deaths, but it could be from this or it could be from something else, right? You don't always know with these things. Same with when COVID happened. We wouldn't necessarily know the exact reason for every single death, but it gives you an estimate. Um, is that it? Is that are we good? No antibiotics used to treat EVD. Is it because it's caused by a virus? Yes. If it's a virus, antibiotics do not kill viruses. They only affect kill bacteria. Just going lastly back to the photosynthesis thing. So as I said, the rate is affected by three things: light intensity, temperature, and carbon dioxide, which is why greenhouses are often used to actually grow most of the things that we eat. Because in a greenhouse, the advantage is that you can do three things. You can keep it warm all day long, all year round, which you wouldn't be able to do outside, certainly in the UK. You can keep it, the lights, you can keep it bright all year round, all day long, which you can't do anywhere because you can't do outside because it gets nighttime, right? So there's no light. So by having it all indoors in a greenhouse, you can turn the lights on at night. So you can keep the light intensity high all the time. You can keep the temperature right all the time. And you can also keep the amount of carbon dioxide high all the time inside. How could you do that? Well, if you think about burning fossil fuels, what does that release? It releases carbon dioxide. So all you do inside this greenhouse is you have like in the corner, like a little heater going, like a little fire burner, which obviously keeps it warm a little bit as well. But crucially, if you're burning a fuel, it releases carbon dioxide. If I wanted to increase the amount of carbon dioxide in the room that I'm in right now, I could just have a little fire going in the corner with safety precautions in place. Um, and it would just be adding carbon dioxide to the room as well as obviously heating it up. So we can do all three of those things. We can keep all three of the factors that affect the rate of photosynthesis really nice and high indoors in a controlled greenhouse. There are other advantages of a greenhouse as well, which is that these things are not going to be eaten by, I don't know, a rogue fox roaming around because we've got doors and it's closed. Diseases are less likely to spread in a greenhouse because it's all under your control. You can put the exact amount of water down, whereas outside, it can rain too much or rain too little and it gets all really complicated. Um, and that basically, but the problem with the greenhouse is it is expensive and therefore that is a problem. Is that everything? Are we done? Can I go home? Um, Yeah, it seems like there's nothing else. So, in that case, that is it. Next, just so you know, for the remaining few lessons that we have, we are closed from the 17th of July. You will have like a chemistry revision lesson, a physics one, a maths one, and then at the beginning of July, you will have a test. And then in the second lesson of July, you'll just go through the test, and that will be the end, and we can all enjoy the summer holidays. So that's it. I'll